Welcome to our lecture on spermatogenesis. Imagine a highly specialized factory in the human body whose sole purpose is to manufacture millions of tiny, swimming delivery vehicles every single day. These vehicles carry half of a human's genetic blueprint. Today, we are going to explore this incredible biological assembly line, understanding how a basic, unspecialized cell transforms into a sleek, microscopic torpedo. This process is the very foundation of how human life is passed from one generation to the next.
To begin, let us look at where and how this manufacturing takes place. Spermatogenesis is the complex biological process where male primordial germ cells, known as spermatogonia, undergo division and differentiation. This entire sequence happens within the tightly coiled seminiferous tubules located in the testes. It is a continuous, highly organized process that never truly stops. Understanding this is critically important for medicine, particularly in diagnosing and treating male infertility, developing contraceptives, and addressing reproductive health issues. If this delicate assembly line is disrupted by toxins or heat, the factory shuts down. Now that we know where this happens, let us break down the assembly line into its three distinct phases. First is spermatocytogenesis, which uses mitosis. Here, the stem cells multiply to ensure a never-ending supply of raw materials. Next is meiosis, a crucial step where the genetic material is halved, ensuring the resulting sperm contains exactly one set of chromosomes. Finally, we have spermiogenesis. This is the physical remodeling phase. The round cells shed excess cytoplasm, condense their DNA, and grow a long flagellum, or tail, equipping them with the motor they need for swimming. Understanding these phases allows us to look at the efficiency of this biological factory mathematically. Because the process relies on meiosis, a single starting cell undergoes 2 rounds of division to maximize output. We can express this total yield with a simple equation: y equals p times 4. Here, y represents the total yield, or the final number of mature spermatozoa. P represents the initial number of primary spermatocytes that successfully enter the first meiotic division. The number 4 is a biological constant, representing the 4 haploid cells generated from one diploid cell. If the starting pool, P, increases, the total yield, Y, increases proportionally by a factor of 4. Let us see this formula in action with a concrete example. Suppose a medical researcher is studying a small cross-section of a seminiferous tubule to evaluate a patient's fertility. She counts exactly 250 primary spermatocytes preparing to undergo meiosis. We want to know how many mature spermatozoa will be produced. We start with our yield formula, y equals p times 4. We substitute our starting value of 250 for p. Multiplying this by our biological constant of 4 gives us 1,000. This means that just 250 stem cells will successfully develop into 1,000 mature spermatozoa, demonstrating the massive amplification power of the male reproductive system. Now that we have used this formula, let us understand exactly where that biological constant of 4 comes from. The multiplication step represents the biological reality of the 2 meiotic divisions. When a primary spermatocyte enters meiosis, it first undergoes meiosis 1. This initial division splits the primary cell into 2 secondary spermatocytes. But the process does not stop there. Each of those 2 secondary spermatocytes immediately undergoes meiosis 2. This second division splits them again, resulting in 4 spermatids. These 4 spermatids then mature into 4 spermatozoa. This perfectly captures how one single cell divides twice to create a quartet of new, genetically unique cells.
This brings us to another critical aspect of spermatogenesis, tracking the genetic content. As these cells divide, their chromosome numbers change. A normal human spermatogonium is diploid, meaning it has a full set of chromosomes. When it grows into a primary spermatocyte, it remains diploid. However, the first meiotic division is a reductional division. It cuts the chromosome number exactly in half to create secondary spermatocytes. The second meiotic division is an equilational division, meaning the chromosome number stays the same for the spermatids and the final spermatozoa. This precise reduction is absolutely critical for human reproduction. To find the exact chromosome numbers at the end of this process, we must understand the mathematical reduction that occurs during meiosis. We can express this with the formula n equals 2n divided by 2. In this expression, the term 2n represents the diploid chromosome number of the primary spermatocyte. The variable n represents the final haploid chromosome number found in the secondary spermatocytes, spermatids, and mature spermatozoa. We divide the starting number by 2 because the homologous chromosome pairs are separated during the first meiotic division. If the diploid number changes, the haploid number will always be exactly half of it. Before we move on, let us see this with a concrete example using human genetics. A biology student wants to know the exact number of chromosomes in a mature human spermatozoan. We know that a normal human primary spermatocyte starts with 46 chromosomes. This is our 2n value. To find the haploid number, we use our reduction formula. N equals 2n divided by 2. We substitute 46 into the numerator. Dividing 46 by 2 gives us exactly 23. This calculation reveals that a secondary spermatocyte, and ultimately the final mature spermatozoan, contains exactly 23 chromosomes. So we have proven that the haploid number is 23. This brings us to why this mathematical reduction is biologically necessary. During the first meiotic division, homologous chromosome pairs are physically pulled apart. This division ensures the resulting cells only get half the genetic material. It is a necessary mathematical reduction to prevent the chromosome number from doubling every single generation. Because the subsequent division and physical maturation do not change this number, the sperm carries exactly 23 chromosomes. When it fuses with an egg, which also carries 23, it perfectly restores the normal human number of 46 chromosomes in the new baby. Let us bring all of these concepts and formulas together by looking at this diagram of the entire assembly line. At the very top, we start with the diploid spermatogonium, which grows into the primary spermatocyte. Notice the label 2n, indicating it contains a full set of chromosomes. Following the arrows downward, you can see the first meiotic division splitting this cell into 2 secondary spermatocytes, now labeled with a single n for haploid. The next branching shows the second meiotic division, creating 4 round spermatids. Finally, at the bottom, we see the result of spermiogenesis. The 4 spermatids have physically remodeled into sleek, swimming spermatozoa, ready to deliver their genetic payload. To wrap up our lecture, let us review the key takeaways. Spermatogenesis is a remarkable biological assembly line that begins with diploid stem cells and utilizes the mathematical precision of meiosis to produce 4 haploid spermatozoa from every primary spermatocyte. We saw how this process not only ensures a massive and continuous supply of sperm, but also guarantees that each sperm carries exactly half of the genetic information needed to create a new human life. By understanding these stages of cell division and physical remodeling, you now have a deep insight into the foundations of human fertility, genetics, and reproductive health. Great job following along.